Blood leaves the heart and flows into large arteries where pressure is high. These arteries branch into narrower and narrower arteries and then into arterioles until finally becoming capillaries. Each capillary is made of a single layer of endothelial cells with tiny pores between them. The pressure inside the capillary pushes water and small solutes out of the blood through these pores. The fluid that leaves the capillary moves into a tiny space between tissue cells. This area is called the interstitial space and is where exchange with the cells take place. Every day, about 20 liters of fluid move out of the capillaries and become part of the interstitial space between the cells. About 17 liters gets quickly reabsorbed back into the capillaries, but that leaves about 2 to 3 liters of fluid behind in the tissue each day. This 3 liters of fluid needs to find a way back into the blood so that the body's interstitial fluid volume and the blood volume both stay constant over time. That's where the lymphatic vessels come in. They collect excess interstitial fluid and return it into the blood. If this return system fails or becomes blocked, fluid begins to build up in the tissues, a condition known as edema. So in this video, we are going to go through everything you need to understand this system fully, starting with the 4 main functions of the lymphatic system. After that, we will follow the lymphatic vessels and see how they're arranged in the body. Then, we will look at the structures that make up this system, divided into the primary or central lymphoid organs, which are the thymus and the bone marrow, and the secondary or peripheral organs, the spleen, tonsils, lymph nodes, and the lymphoid nodules. And along the way, I will also mention a few clinical conditions related to this system. What's up everyone, my name is Ty. I'm a medical doctor and I make animated medical lectures to make different topics in medicine visually easier to understand. If you would like a PDF version or quiz of this presentation, you can find it on my website, along with organized video lectures to help with your studies. Alright, let's get started. So, what are the functions of the lymphatic system? This is honestly such an interesting system once you understand how it works. The first function is extracellular fluid drainage. Remember, about 20 liters of fluid gets filtered out of the blood into the tissues every day. About 17 liters gets reabsorbed straight back into the blood, and that leaves about 2 to 3 liters behind in the interstitial space. Now, this system is really important to understand properly, so let's take a closer look at how this actually happens. Let's start by zooming into the capillary here. Blood flows from the arterial side of the capillary towards the venous side. Inside the capillary, there is blood, and blood is a fluid. This fluid exerts a force on the capillary wall, and this force tends to push fluid out of the vessel and into the surrounding tissue space. Because the blood arriving at the arterial end is under higher pressure, this outward direction force is stronger at the arterial end. As the blood travels through the capillary towards the venous side, this pressure gradually decreases. Outside the capillary, in the interstitial space, there is also fluid, but the pressure here is much lower than the inside the vessel, so the net movement of fluid is outwards. This outward pushing force generated by the fluid is called hydrostatic pressure. There's also another important force working on the opposite direction called colloid-osmotic pressure or oncotic pressure. Now, the plasma inside the capillary contains proteins, mainly albumin, which are too large to pass through the capillary wall under normal conditions. These proteins attract water and create a pulling force, drawing fluid back into the capillary. The interstitial space contains far fewer proteins, so the pulling force from the outside is much weaker than the one inside the vessel. And since the force pulling the fluid inwards is higher, the net pressure along the capillary is like this. At the arterial end, the hydrostatic pressure pushing fluid out is stronger than the colloid-osmotic pressure pulling fluid in, so more fluid is going to leave the capillary here. As the blood continues towards the venous end, the hydrostatic pressure gradually falls because energy is lost due to resistance along the capillary. The colloid-osmotic pressure, however, stays relatively constant along the capillary since the concentration of plasma proteins remain unchanged. This means that towards the venous end, the inward pulling force becomes stronger than the outward pushing force, allowing some of the fluid to return back into the capillary. Even with this reabsorption, the amount of fluid that enters at the arterial end is still slightly greater than the amount that returns at the venous end, and over time, this produces a small but continuous accumulation of fluid in the interstitial space. If this excess fluid was not removed, it would gradually build up and cause swelling, known as edema. This is where the lymphatic system becomes essential. It collects that extra fluid from the interstitial space and returns it into the bloodstream to maintain normal fluid balance. Let's zoom into the lymphatic capillary to see how the lymphatic system actually does this. At the very beginning of the lymphatic network, we have the lymphatic capillaries, and all of this surrounding them is the interstitial space. Unlike the blood circulatory system, the lymphatic system is not a closed loop. It's an open-ended system that begins in the tissues. These lymphatic capillaries are the smallest lymphatic vessels, and they are structurally designed to be highly permeable. Their walls are formed by a single layer of endothelial cells that loosely overlap each other. This overlapping arrangement forms small one-way mini-valves. When the pressure in the interstitial space becomes higher than the pressure inside the lymphatic capillary, these mini-valves open, allowing interstitial fluid to enter. Once the fluid moves inside and the pressure within the lymphatic capillary rises above the surrounding tissue pressure, the mini-valves close, preventing fluid from leaking back out. This ensures a unidirectional flow of fluid into the lymphatic system. To help maintain their structure, lymphatic capillaries are anchored to the surrounding tissue by collagen filaments. These anchoring filaments prevent the walls from collapsing when the interstitial pressure changes, allowing the capillaries to remain open and functional. The fluid that enters the lymphatic system is called lymph. In most tissue, lymph is very similar to interstitial fluid, consisting mainly of water, dissolved ions, and small amount of plasma proteins that have escaped from the blood capillaries. There is, however, one important exception in the small intestines. Here, specialized lymphatic capillaries called lacteals absorb dietary fats. These fats are packed into chylomicrons and enter the lymph, giving it a milky appearance known as chyle. This means that in addition to draining extracellular fluid, the lymphatic system also transports larger molecules that cannot easily enter the blood capillaries, such as plasma proteins and lipids. This is the second major function of the lymphatic system. On average, the body returns about 2 to 3 liters of lymph to the bloodstream each day. All of this lymph eventually flows towards the heart and enters the large veins at the root of the neck, bringing the recovered fluid back into the circulation. Another important function of the lymphatic system is removal of old or damaged cells. A key organ to this is the spleen, which filters the blood and removes aged or abnormal red blood cells. The fourth main function of the lymphatic system is immune surveillance. As lymph travels through the lymphatic vessels, it passes through multiple lymph nodes. These lymph nodes contain large population of immune cells that monitor the lymph for pathogens such as bacteria, viruses, or abnormal cells. If anything potentially harmful is detected, the immune system is activated. We will look at this process in detail later in this video, but for now, those are the 4 core functions of the lymphatic system. Let's tick that off and now continue with the lymphatic vessels, step by step, so the entire system becomes clear and logical. Now that we have seen how the lymph is collected at the tissue level, let's place this structure in context. At the very beginning of the lymphatic system, we have these blind-ended lymphatic capillaries that contain one-way valves and take in fluid based on pressure differences. They are located right here. Once they take up fluid, these capillaries merge and gradually form larger and larger lymphatic vessels as they move towards the venous system. So the progression is lymphatic capillaries, then lymphatic vessels, then lymphatic trunks, and finally lymphatic ducts, which return lymph to the bloodstream. Lymphatic capillaries are blind-ended tubes, so this open-ended structure ensures a unidirectional flow of extracellular fluid. They collect fluid along with metabolic substances, antigens such as bacteria, viruses, or even tumors or pre-tumor cells, as well as antigen-presenting cells. These capillaries form interconnected networks that run alongside blood capillaries and venules. In the small intestine, the lymphatic capillary run within the core of each intestinal villus to allow absorption of dietary fats. From the lymphatic capillaries, lymph drains into larger lymphatic vessels. These vessels are thin-walled and contain valves that maintain forward flow, preventing backflow of lymph. Because they collect lymph from the tissues, these are often called collectors. Within organs, we call them subcapsular and deep lymphatic networks, but in the limbs, lymphatic vessels are divided into superficial and deep collectors that follow specific veins. In the upper limb, the superficial collectors are grouped into three main pathways. The lateral collectors run along the cephalic vein and drain into the axillary lymph nodes. The medial collectors run along the basilic vein and also drain into the axillary lymph nodes. The anterior collectors run along the median antebrachial vein and drain into the superficial cubital lymph nodes. In the lower limb, the superficial collectors are grouped as the medial collectors, which follow the great saphenous vein and drain into the superficial inguinal lymph nodes. The lateral collectors, which follows the tributaries of the great saphenous vein and also drain into the superficial inguinal lymph nodes. And the posterior collectors, which accompany the small saphenous vein behind the knee and drain into the superficial popliteal lymph nodes. The deep lymphatic vessels run alongside the deep veins of the lower limb and drain into the deep popliteal and deep inguinal lymph nodes. So, lymphatic capillaries drain into lymphatic vessels, and these then drain into the lymphatic trunks. The lymphatic trunks are larger channels that drain lymph from specific anatomical regions. Most of these are paired, meaning there is a right and left trunk. The jugular trunks drain lymph from each half of the head and neck, the subclavian trunks drain lymph from the upper limbs, and the bronchomediastinal trunks drain lymph from each half of the thorax. On the right side, these three trunks unite to form the right lymphatic duct, which is a short duct that collects lymph from the right side of the head and neck, the right upper limb, and the right half of the thorax, and drains into the venous system at the junction of the right subclavian and the right internal jugular vein. In the abdomen, we have the lumbar trunks, which drain lymph from the lower limbs and the pelvic region. We also have a group of unpaired trunks that drain lymph from the abdominal organs. These trunks converge into dilated sac called the cisternoculi. From here, lymph travels upwards through the thoracic duct. The thoracic duct is the main lymphatic channel in the body. It receives lymph from the rest of the body and drains into the venous circulation at the junction of the left subclavian and the left internal jugular veins. So, as you see here, lymph flows from the lymphatic vessels, passes through several lymph nodes, and as it continues towards the heart, these vessels unite to form lymphatic trunks, which then merge into larger lymphatic ducts. That was the lymphatic vessels, so let's tick that one off as well. Now what I want to do is skip all of these topics for now and start with the lymph nodes. What they are, how they work, and why we have them in the first place. We will then continue with the primary and the secondary lymphatic organs so it becomes logical. As lymph flows through the lymphatic vessels, it eventually passes through small, specialized organs called lymph nodes. Imagine for a second that a pathogen, like a virus or bacterium, gets absorbed into the lymphatic capillaries. It then travels with the lymph through the vessels and eventually enters a lymph node, where it will be examined and filtered. Let's see how. If we isolate one lymph node and look at its internal structures, we will see that each lymph node is a small oval organ, usually between 1 to 25 millimeters in size. The human body contains roughly 600 lymph nodes in total. Each lymph node is enclosed by a dense connective tissue capsule that extends inwards as thin partitions called trabeculae. Beneath the capsule lies the cortex, which contain lymphatic nodules rich in B lymphocytes. Deeper to it is the paracortex, which is dominated by T lymphocytes, and at the center lies the medulla, containing medullary cords and sinuses. On the concave side of the lymph node is the hilum, a region where blood vessels enter and exit, and where one or sometimes more efferent lymph vessels carry lymph away from the node. Now, how does lymph actually circulate inside a lymph node? Lymph enters through multiple small vessels called afferent lymph vessels. Once inside, it flows into a space just beneath the capsule known as subcapsular or marginal sinuses. From here, lymph moves deeper through the cortical and the paracortical regions via small internodular sinuses, then into the medullary sinuses located in the center of the node. After being filtered, the lymph exits the node through the efferent lymphatic vessels at the hilum and continues along its pathway towards the larger lymphatic trunks and ducts. Remember, one of the lymph node's main function is immune surveillance. And it's able to do that because inside the node we will find three major types of immune cells: macrophages, dendritic cells, and lymphocytes. Macrophages are found in abundance in the subcapsular sinuses, and they're also present in the medullary sinuses. When lymph enters the lymph node, it also carries with it bacteria, viruses, or small foreign particles from the tissues. As the lymph first arrives in the subcapsular sinus, macrophages act almost like filters. They trap, engulf, and destroy many of the microbes right at the entry points. Dendritic cells are another key player. Some of these dendritic cells picked up pathogens out in the tissues before entering the lymph vessel, and they arrive in the node carrying those antigens with them. Their job is not mainly to destroy the pathogen, but to process it and present small fragments of it, called antigens, to lymphocytes deeper inside the node. The third group of cells here are the lymphocytes. In lymph nodes, lymphocytes exist mainly as B cells and T cells. B cells are found primarily in the cortex inside structures called the lymphatic nodules or follicles, while T cells are located mainly in the paracortex, just beneath the cortex as you see here. Now this is where the immune response begins. After dendritic cells arrive, they migrate into the paracortex. Here, they display those antigen fragments on their surface to T cells. This is why macrophages and dendritic cells are referred to as antigen-presenting cells, because they present antigens to lymphocytes to activate them. When a naive T cell encounters a dendritic cell presenting an antigen that matches its receptors, that T cell becomes activated, and it begins to divide and produce many identical copies of itself. Some of these activated T cells differentiate into helper T cells, which will assist other immune cells, and other become cytotoxic T cells, which directly kill infected or abnormal cells. At the same time, B cells in the cortex can bind directly to antigens that happen to be present in the lymph. They internalize the antigen, process it, and then present it to helper T cell at the border between the cortex and the paracortex. With help from these T cells, the B cells also become activated and start to multiply. Some of these activated B cells move deeper into the lymph node and form circular structures called germinal centers. From here, some B cells become plasma cells, which start producing antibodies and releasing them into the circulation, while others become memory B cells that remain in the body long term, ready to respond quickly if the same pathogen returns in the future. So that's the general idea of how lymph node filters lymph and activates the immune response. Now, let's talk about B and T cells for a moment, because understanding where they come from and where they mature makes the rest of the lymphatic system much easier to understand. Our lymphatic system contains two categories of lymphoid organs: primary or central lymphoid organs and secondary or peripheral lymphoid organs. Primary organs are the places where lymphocytes are produced and where they mature before being exposed to any antigens for the first time. These organs are the bone marrow and the thymus. B cells are produced in the bone marrow and they also mature there. T cells, however, are produced in the bone marrow and then travel to the thymus, where they undergo their maturation process. So B for bone marrow and T for thymus. Once B and T cells have matured, they enter the bloodstream and circulate towards the secondary lymphoid organs, which are the spleen, the tonsils, the lymph nodes, and the lymph nodules found in mucosal tissue. These are the places where lymphocytes finally meet antigens for the first time. And this is where they get activated and actually start doing their immune functions. Let's go through the main secondary lymphoid organs one by one, starting with the spleen. The spleen is the largest secondary lymphoid organ. Its main job is to filter the blood, not the lymph. So unlike lymph nodes, which monitor lymph, the spleen monitors the blood directly. One of its important functions is removing old or damaged red blood cells from the circulation. But in addition to that, it also plays a major role in starting immune responses against antigens that appear in the bloodstream. I do have a detailed video on the spleen anatomy if you want to know more, but the spleen sits in the upper left part of the abdomen, under the ribcage, and is covered by a layer of peritoneum. If we take the spleen, cut it open, and look inside, we will see a thick fibrous capsule on the outside. This capsule sends inwards extension called splenic trabeculae, which divides and support the inner tissue. Inside, we can clearly see two main types of tissue: the red pulp and the white pulp. The red pulp is involved in filtering the blood and removing old or damaged red blood cells. The white pulp, on the other hand, is rich in lymphocytes and is responsible for initiating immune responses against any blood-borne pathogens that enters the spleen. So that was the spleen. Now let's move to the tonsils. The tonsils form a protective ring of lymphoid tissue at the entrance of both the respiratory and the digestive tracts. This means that they act as one of the very first immune barriers for anything we breathe in or swallow. Tonsils are made up of lymphoid nodules, and many of these nodules contain germinal centers where active immune responses take place. We have several tonsils in the throat region. The one most people are familiar with is the palatine tonsil, which sits on each side at the back of the mouth. We also have the inguinal tonsils at the base of the tongue, the tubal tonsils located near the opening of the auditory tubes, and the pharyngeal tonsils, also known as the adenoids, located in the roof of the nasopharynx. When tonsils become inflamed or enlarged, such as during infection, they can become tender and swollen, which is a sign that immune cells inside them are actively fighting off pathogens. So that was the tonsils. Earlier, we already talked in detail about how the lymph nodes function, but now let's briefly look at them from a clinical perspective. Lymph nodes in the body are grouped according to their region. This is important because during a physical examination, doctors often check specific areas where lymph nodes lie close to the surface. If lymph nodes in a particular region are enlarged, it often indicates that there is an infection or inflammation or sometimes something more serious in the area that the lymph nodes drain. For example, in the head region, we have facial nodes, parotid nodes, mastoid nodes, and occipital nodes. In the neck, we have submandibular nodes and the deep lateral cervical nodes. Under the armpit, we find the axillary nodes, and so on throughout the body. I will go through these in more detail in the written PDF version of this video, so you can learn which areas they drain. Now, sometimes lymphoid tissue does not form large organs like a lymph node or spleen. Instead, it appears as small scattered clusters. These are called lymphoid nodules. Lymphoid nodules are small islands of immune tissue that sits within the mucosa of organs. Together, they form what we call the mucosal immune system, or MALT, mucosa-associated lymphoid tissue. This system is extremely important because our mucosal surface, like the gut and respiratory tract, are major entry points for pathogens. MALT can be found in two general forms. There are diffuse MALT, which is made of scattered immune cells within the mucosa. And then there is organized MALT, which forms more visible and structured nodules. Organized MALT includes regions such as GALT, or gut-associated lymphoid tissue, found in the intestines, or BALT, or bronchus-associated lymphoid tissue, found in the airways. There are also others, such as those found in the urinary tract, the conjunctiva, and even the skin. So those were the secondary lymphoid organs. These are places where B and T cells finally encounter pathogens, interact with each other, and becomes activated to start the immune response. And that wraps up our detailed overview of the lymphatic system. I really hope this helped make everything clearer and more visual for you. I've made free courses for other topics here on YouTube if you want to keep learning. Otherwise, if you want a handmade PDF version of this lecture, take a quiz to test your knowledge, or access an organized list of all my videos, you can find everything on my website. Thanks for watching, see you in the next one.